So it's our fifth session of this, and it should say JCBI sent there. We saw it. Okay. I am pleased to announce that there are now more than 90 groups who are either actually, as in you have cells hopefully in a freezer or a refrigerator or an incubator somewhere in your lab, or computationally working with the cell. We added, I think, another 12 or 13 groups last year. Since our paper first published in 2016, we have had now more than 70 papers in the literature that focus on the JCBI minimal cell in one fashion or another, and that is spectacular. And one of the things that I am pleased, and I think is a consequence of this workshop, is that a lot of these papers have authors from a few of these papers have authors from multiple institutions, and they don't include the JCBI, which means that we are not the node of everything that happens now in the minimal cell universe, and that this is a larger effort that doesn't just depend on us. A couple of the updates I wanted to offer you that are near and dear to my heart come back to this reason that we started this project at the JCBI more than 20 years ago. So what we were hoping to get at the question of is this idea of what is life, which is an old question that has haunted chemists and physicists and biologists for more than a century now. But what we're hoping to do using our minimal cell is through the lens of genomics, so we can understand this question of what genes are necessary for life, or maybe what are the tasks that a living cell must perform. And so my team, we sat around, this was led by Chuck Merriman. We sat around a while back and made a list of things that we thought cells had to be able to do. And for instance, they have to have amino acids, cofactors, and be able to do these processes. Now, for instance, we say that a cell has to have lysine. E. coli can make lysine. JCBI, SYN 3.0, etc. They need one protein to bring lysine into the cell. Either of those methods accomplish the task. And so it doesn't matter what genes enable the completion of the task. We just want to know what the tasks are. And so I know you guys should have seen this slide many times, where we did a blast search of the SYN3A, the SYN3 genes. And so if we blasted the SYN3 proteins against E. coli, everywhere where a minimal cell gene hit a gene in E. coli, you get a colored bar. But if there's no similar gene in E. coli, you get a white bar like there. And so we divided this into either genes we know what they do, either equivalent, meaning that there is a tiger fam that matches this gene. And remember, no new tiger fams have been made in the last 15 years. Or we're really, really confident of a cell's function. So here you see a great deal of color in the genes where we are really quite sure we know what the protein does. We've got a smaller number of putatives, and then we've got these generics and unknowns. Now, over time, in 2016, there were 149 genes we didn't know what they did. We have now looking using a variety of methods and say that there are only 66 essential genes in our minimal cell that encode proteins of unknown function. I don't count in that list the 78 non-essential genes in the minimal cell. A lot of those are mycoplasma-specific genes and are of unknown function, but I'm not so interested in those. I want to know the things that the cell has to do, and I'm hoping that we can collectively, as we move forward in this process, that a number of labs in this consortium that we have will start thinking about this question. So as I said, there are only 66 genes of unknown function. 35 are unknown unknowns. 31 are generic unknowns that we know they're a hydrolase or a transporter, but we don't really know what their substrates are, their reaction purposes. A lot of these genes have orthologs, almost all of them have orthologs in almost everything. Liam Chiteak, an MIT graduate student I work with, is building me a new version of that last slide I showed you, where we reduce the stringency of the blast. It is still a highly confident blast, but we made it a little too stringent in 2016. And then it's also worth noting that this needs to be done. So as some members of our consortium are thinking about designing new synthetic genes, these are really important considerations. I've also, because of this workshop and the efforts of James Dobbinspec, who we're going to hear from in a little bit, we know that it is likely, because of protein moonlighting, the number of tasks a cell must perform may be much greater than the number of essential genes. So, for instance, computational analysis of Syn3A genes identified 98 proteins with more than one functional domain. That's just a partial list. That's Liam at MIT who did this work for me. And then James Dobbinspec, and James, this is the only photograph I can find when I think that's your high school picture, probably. So James has shown that there are 28 minimal cell proteins that are located, whose proteins are located at more than one cellular site. And so, again, thinking about this question that has been driving me, among many, my group, is how many tasks must a living cell perform? It is likely that of the 400 essential genes the cell encodes, there may be a lot, and there may be more than 100 with moonlighting functions. So the total number of tasks the cell must perform could be way over 500. And these facts need to be considered as we move closer and closer to an era where we start building living synthetic cells.